Hey, so we did the POLH gene translesion synthesis and its clinical relevance. So what is translesion synthesis? Translesion synthesis is a DNA repair mechanism that allows replication to continue past a damaged piece of DNA. Humans need this because it's the way we handle UV radiation-mediated DNA damage. And the way this works is multiple specialized error-prone DNA repair polymerases are stored in a subnuclear compartment for when the replication fork stalls at a lesion. The normal DNA polymerase is replaced by one of the specialized polymerases so it can bypass the damage and extend to a suitable position downstream where another polymerase change will occur in which the normal high-fidelity polymerase will replace the specialized polymerase. Without this tolerance, damaged DNA would block replication from continuing, invoking cell death. And on the left, you see an image that describes this mechanism in which the normal polymerase is exchanged with the TLS polymerase so it can skip over the cell damage, or the DNA that's damaged, and then be re-replaced by the normal polymerase.
Now let's move into the specifics of the POLH gene. So this particular gene codes for the DNA polymerase eta. That's one of those specialized TLS polymerases that Connor was talking about. So in particular, this focuses on doing translesion synthesis over UV-created pyrimidine dimers. It does some other ones as well, but it focuses on those UV dimers in particular. Just like he was talking about, it's going to insert one or two different nucleotides that are opposite from that particular lesion, and that allows an entirely new complex, the POLZ complex, to then come and extend the primer over top of it so it can bypass that DNA damage without having to do any sort of direct repair, given that human beings don't have any direct DNA repair mechanisms for this particular break. In addition to working with translesion synthesis, it also helps with base excision repair. It actually forms a shift-base pair with one of the enzymes involved during the A-basic site, and as a result, it actually inhibits DNA synthesis during that time while it's performing repair.
Xeroderma pigmentosum is a disease that is caused by the POLH disruption that we talked about earlier. When these things go wrong, like we talked about earlier, the UV dimers is one of the main things that POLH fixes, or the polymerase that's encoded by the POLH gene fixes. Xeroderma pigmentosum, it presents with several symptoms related to that, including ocular degeneration, extreme sunlight sensitivity, like second-degree burns within 15-30 minutes in the sun, extremely early onset of skin cancers, such that the majority of patients have cancer before the age of 10, and premature aging, along with neurological abnormalities, although those are typically more prevalent in the other variants. The XPV accounts for 30% of the xeroderma pigmentosum cases, so it is a hefty portion of these patients. This version, like I said, doesn't really have the neurological abnormalities, although it still can, although they're typically not as good. The other versions are typically caused by defects in nucleotide excision repair, whereas this one is a little different because patients have intact nucleotide excision repair, but they are deficient in the ability to bypass the UV-induced DNA damage during DNA replication. So the diagnosis typically happens through clinical evaluation and genetic testing. There's really no cure for this disease. The best you can do is stay out of the sun, and the prognosis is unfortunately not really amazing. Although it is better for people with the variant or POLH disruption than it is for other varieties, with a life expectancy in the late 30s to early 40s rather than in the early 30s. The best way that we can prevent xeroderma pigmentosum is by genetic testing. Both parents should get genetically tested for all varieties of the disorder, especially if they have a family history. And early diagnosis and intervention are critical for the people who have this disease to live the best life that they can.
So let's think about this abstractly. If you are a family that is having this problem because it's autosomal recessive, what do you do about that? There is a Guatemalan village that had considered this to be a curse among certain families where they were having all of this skin damage just from this exposure to the sun, as Jimmy was explaining. And they didn't know what to do about it. Eventually, a missionary had come in that was working for a medical organization and saw these kids with all these problems and tried to figure out what's going on. And long story short, they found out that it was xeroderma pigmentosa. And so the issue is that even though they had this diagnosed eventually, and they started realizing that it's running through families, there's no good treatment for it. The prevention is the best method in the sense of you're trying to get a lack of exposure to UV. You're trying to stop yourself from having continual UV exposure. But after that, there's not any good treatments for that. And so that's where we're going to look at some recent developments. So to solve the problem, we have to understand the problem. That's the importance of understanding the different factors of the way that UV damage or UV radiation causes damage to the genetic material within the cells. And so one abstract that we saw showed that not just is this causing chromosomal damage, but it's also causing mitochondrial damage. How does that relate to what we've discussed? So we saw that this disease can also cause neurodegenerative issues, and that's what they're thinking is causing this. They're thinking that mitochondrial damage is causing these neurodegenerative issues. And so it might not be a primary role within xeroderma pigmentosa, this mitochondrial damage, but it leads to further issues within the rest of the body, which then just shows that this is not something that's isolated to the skin, but it shows how this can impact other parts of the body. So then how does that affect the way in which we come up with treatments? Currently, the best way that they have to treat this is removing these portions of the skin, just like you would have with malignant melanomas, and that you just cut out the skin and hope it doesn't spread. But in this case, they want to try to find out a better way to treat that, because this is not something that's just with a singular spot within the skin. This is throughout the body that's having these issues because it's not able to fix the damage that comes about from the UV radiation. So one of the treatments that they saw that had some promise was from a form of DNA repair that comes from bacteria. We've already seen that with CRISPR-Cas9. We can target genetic portions of different chromosomes or capsids, but here they're using a specific repair enzyme called T4N5 endonuclease to be able to target the different forms of genetic damage, specifically ones that they're seeing cause damage. In other words, we're basically seeing that this is able to treat different diseases that are similar to ours, such as actinic keratosis and basal cell carcinomas, issues within the skin. And so they're hoping that this can also then cause the same sorts of improvements within xeroderma pigmentosa. But it's not as simple as we'd like it to be because of all these different damages, pathways to the DNA.
And last but not least, we got a couple of questions for you guys. Question 1 is, why do we care if DNA synthesis is slowed down during base excision repair? And second, we want to ask, why are treatment options difficult to create for xeroderma pigmentosum? Thanks for listening, and we'll see you guys in class.